Greetings and welcome to your first fully vodcasted lecture of the semester. There are a few announcements that I have for you, and if you're watching this from during the regularly scheduled lecture day, then today is February the 5th, though I imagine that all of you are watching this at your leisure and in random places. So let's make a few announcements. February the 10th is when we would really like you to get your group members' names, your group name, and your tentative topic emailed to me. So with this, please help me out by sending both the first and the last names of your group members so that I make sure that I find the right person, because sometimes we do have somebody with the same first name and then I confuse them with someone with the same name and different last name. So send me first and last name, that's great. And then if you could send me your group name as well, hopefully you've come up with something awesomely creative, and a tentative topic. This can be very broad right now. It needn't be the ultra narrow and refined topic that it will eventually become. For example, you could just tell me that you're planning to do your project on methicillin-resistant Staphylococcus aureus, and right now that would be enough. However, with time, we're going to see that funnel in scope, and when you make a hypothesis, of course, it will become very focused. Your second homework is due on Tuesday, so look towards that. Thank you for your patience in my grading homework 1. That will take me a few days to get done. So it just is, it's a hoof. It takes like 8 hours to get through everybody's. So also remember that today, if this is the regularly scheduled lecture day for you, I will be holding a macromolecule review. We'll finish off some of the additional review questions in the macromolecule game that we didn't get to. And we'll also do a little bit of practice with questions, kind of like we did the other day. And I'll also hold office hours. So I will literally be online. And let me make sure that I add this on here. So this will be from 11 a.m. to 1 p.m. So a full 2-hour time frame that you can catch me on the conferences feature on YO courses, February the 5th. If you're watching this again on the regularly scheduled lecture day, that's today. So I look forward to seeing, I hope, a lot of you on that session. Let's go ahead and get started today. We want to begin by looking at the 3 domains of life. There, of course, is that which we are most familiar with, the eukarya. We are like the fungi and the protists, and actually, I'm going to jot that down. So the eukaryotic world, of course, encompasses us, but it also encompasses a lot of microorganisms. The microorganisms that are a part of the eukaryotic world are the fungi and also the protists. So if we were to divide it into 2 major microbial categories, we would have the fungi and the protists. And then it's cool to note that these 2 categories can be further broken down. So within the fungal world, the fungi among us, we have the yeast and the molds. And then the protists could also be further split into the algae, quote unquote, and the protozoa. Now, the reason that I say quote unquote for these is because phylogenetically, which means based upon DNA sequences, these are not really legitimate groups. However, when we think about one being the light-using photosynthetic protist versus one being the eaters, the consumers that eat and chow down on other life, we split them into those 2 groups for that reason. We'll talk more about that later, but this gives us the rough idea of the kinds of microorganisms that fit into the eukaryotic domain of life, the domain eukarya. Now, there are 2 other domains of life, the archaea and the bacteria. The archaea, rightly so, are their own domain. They have characteristics of both bacteria and eukarya. Now, by all right, bacteria are the dominant form of life on Earth. Now, I know, wait, check that. Not humans? No, bacteria are the dominant life form on Earth. And I actually want to read to you a sexy-minded passage, and I know a couple of others said that you are very interested in literature. And so let me read to you a passage from a book that I would encourage you to read. In fact, any of Stephen Jay Gould's works are absolutely worth your time in picking them up. This is a passage from Planet of the Bacteria. On any possible, reasonable, or fair criterion, bacteria are, and always have been, the dominant forms of life on Earth. Our failure to grasp this most evident of biological facts arises in part from the blindness of our arrogance, but also, in large measure, as an effect of scale. We are so accustomed to viewing phenomena of our scale, size measured in feet, and ages in decades, as typical of nature. Individual bacteria lie beneath our vision and may take no longer than the time I take to eat lunch or my grandfather spent with his evening cigar. But then, who knows, to a bacterium, human bodies might appear as widely dispersed, effectively eternal, or at least geological, massive mountains fit for all forms of exploitation and fraught with little danger. It's very interesting to think about our bacterial habitants as thinking about our human bodies as those geological features. But undoubtedly, Stephen Jay Gould captures that idea that bacteria are the dominant forms of life on Earth. And in fact, we have cultured and characterized less than 1% of the bacterial world. So this world is full of unknown characters, many of which we're just beginning to elucidate in a genetic way, finding some of their DNA, but not still being able to grow them and to know what they need in order to sustain their pure culture growth. Now, it has been traditional that bacteria and archaea are referred to collectively as the prokaryotes. So you've probably talked in your previous courses about the eukaryotes versus the prokaryotes. My challenge question to you this semester is to think about whether or not the term prokaryote is even appropriate. Should we have it? Because bacteria are very different from archaea, which are very different from eukarya. Archaea are related to both bacteria and eukarya and have features of each. Thus, the term prokaryotes, collectively referring to bacteria and archaea, might be questioned in its appropriateness because in many ways all it does is to say not one of us, right? So I want you to think about this and I want you to go back to it. And at the very end of the semester, I'm hopeful that you will be able to tell me whether or not you think the term prokaryote should be retained. That being said, I am going to tell you about a nuance of spelling. This is not, so if I ever spell prokaryotes with a C instead of a K, it's not because I don't know how to spell, which is sometimes true as well, but it's because that is an alternative spelling. You may see in textbooks either a C or a K used, so that's something that's important to keep in mind. And speaking of nomenclature and how we correctly write about the microbial world, we name bacteria using a binomial system of nomenclature. Binomial, two names, a first name and a last name. These names are a genus name and a species name. Both of these names are underlined or italicized. This is commonly done incorrectly. In fact, in a moment, we're going to look at a news article in which they have neither italicized nor underlined a scientific name. They're wrong. They're incorrect. When you all do this, I want you to challenge yourselves to think about always either underlining or italicizing the names. When you're working on a computer, you would use italicization. If you're working on the chalkboard, for example, you would just go ahead and underline. So here's the first name, the genus name. And a good example is E. coli. That is Escherichia coli. You might think, well, where does that genus name come from? Turns out it comes from the man who discovered this bacterium. His name was Theodore Escherich. So his last name was the root for the genus name. Now, today, by the system of standing nomenclature, we are no longer allowed to name bacteria after ourselves. So if I discover a bacterium, I can't call it the Rachelia, because because for one thing, that would just sound really bad. But for another, it's not allowed. But at the time of Theodore Escherichia, he was able to name the bacterium after himself. We now have oftentimes better and more appropriate names. For example, in a moment, I'm going to show you a picture of a bacterium called Thiomargarita. Well, the thio refers to its ability to utilize sulfur, and that's a much more fitting and appropriate name. So often comes from the name of the discoverer. The second name, coli, is the species name. And this one is very helpful because it tells you from where an organism is isolated. Coli is the colon. Okay, so the intestine, and this is a GIT bacterium. So this is showing us from where that organism was isolated. The one that I just told you about, Thiomargarita, the genus name, its species name is Namibiensis. So you might think that it was isolated in Namibia, right? And it actually was. We'll look at that in just a moment. So the genus name is commonly abbreviated, and you'll get that feeling because you've probably never heard of E. coli called Escherichia coli. In fact, it almost always goes by its abbreviated name. So E. coli is the name that we typically see for this bacterium. This abbreviation is appropriate and it's accurate. However, it should never be used the first time that you refer to a bacterium. So if you're writing a paper and you mention Escherichia coli for the first time, you should write out the genus name first. And then the subsequent times that you use it, you can say E. coli, and that's appropriate. Now, these aren't the only names for a bacterium, a first and last name, so to speak. That is a genus and a species name. But in fact, we often need to differentiate. I remember at one point I was talking to one of my students, and she called home to tell her mom that she was working with Escherichia coli in the lab, E. coli. And her mom really freaked out. She was like, "Oh my God, you're working with that terrible pathogen." Well, the E. coli that we work with in the lab is completely non-pathogenic. Now, I don't suggest this, but you could lick a plate of our E. coli and you would not get sick because it is a non-pathogenic laboratory strain. However, if you were working with E. coli 0157H7, that strain is highly pathogenic. It is the acid-tolerant bacterium that sometimes is found in hamburger. So when we think about strain designation, it's important often to relate that strain, and often we will put that after the species name. So we'll say E. coli K12, for example, which is a laboratory strain of E. coli, or E. coli B, or we'll say E. coli 0157H7, and I'm actually going to jot that down because that is the very pathogenic strain. So E. coli 0157H7 is one that we will never work with in our lab. Okay, I'm trying to underline it there so that's clear. E. coli 0157H7. And make note that that is a 1 right there. Excellent. Now, this is a perfect segue to look at a newspaper article that will allow us to see what they have done wrong with the binomial system of nomenclature. Researchers have detected an antimicrobial compound in fat, that is adipocytes in fat tissue. This is antimicrobial against the bacterium Staphylococcus aureus. So notice here is a picture of Staphylococcus aureus, and as we scan down through this article, you'll notice here that they're talking about Staphylococcus aureus, and they've done two things wrong. Number one, this is the first time they've used the bacterial name, so it should say Staphylococcus aureus. Also, make note that it is not italicized. That is also incorrect. Okay, sorry, I couldn't get rid of the Zulily advertisements. Those are some studly shoes. Okay, getting rid of that. This is also a very nice segue, and actually I'm going to bring back that picture of the Staphylococcus aureus over here to the side. And I want you to take note that Staphylococcus causes staph infections. That is a lingo term for the disease caused by this bacterium, Staphylococcus aureus. Notice that it is a sphere. So the shape of this bacterium is a sphere. And you can also notice that it's clustered, and that arrangement is actually sometimes fittingly called the Staphylo arrangement, or just simply grape-like clusters. So one shape that bacteria can have is the Staphylo shape, or excuse me, one shape that bacteria can have is the Coccus shape. One arrangement that they can have is the Staphylo arrangement. So some bacteria are spheres, other bacteria are rods, and actually I want to introduce you to someone. My prior students at one point named this fella. I can't remember. I think they said they wanted him to be called Norbert or maybe Philbricht. I can't remember. If you guys have a name for him, you can include that on your extra credit too. But I want you to take note that Philbricht is going to be coming into our future conversation. And he is a beautiful bacterial cell. But notice that he is a rod, okay? So very much a rod-shaped bacterium. One of my skiers just came in the door and she said, wow, that is a big rod bacterium you have there. Indeed. So Philbricht will be coming back into the picture here shortly, but I wanted to use him as an example of a bacillus-shaped bacterium. So we have cocci, spheres, bacilli, rods. The cocci can be in different arrangements. One arrangement is the grape-like clusters. Another is diplo, meaning that there are two cocci side by side. Neisseria gonorrhoeae and Neisseria meningitis are two good examples of a bacterium that has a diplo arrangement. Now, if you're up for a good story at some point, have me tell you my Neisseria gonorrhoeae story. No, not personal experience. Well, kind of. I had a really interesting experience with one of my TAs at one point, bringing in a sample. So have me tell you, it involves the FBI, so we'll talk later. Now, the other arrangement that is, some of you are wearing jewelry right now. Maybe you're wearing a string of pearls. This beads on a string arrangement is often termed a strepto arrangement, and that is largely because the genus Streptococcus is one of the genera that forms this beads on a string arrangement, and so that's why sometimes the name is used, strepto arrangement. You have all heard of strep throat. Strep throat is caused by Streptococcus pyogenes. It's one example of a bacterium that does form this chain-like arrangement. The reason for this chaining arrangement is because the bacteria divide in a single plane repeatedly, and that's what gives rise to the long chain of cocci. Now, we just met Staphylococcus, and recall that it had these clusters of arrangement, and in the variable division in different planes is what gives rise to this particular arrangement. Now, it's important to note that although we often call this arrangement a Staphylo arrangement, Staphylococcus is not the only genus that has this arrangement. In fact, it is often observed for the genus Micrococcus as well. And the same thing is true of the chaining arrangement. We often call it a strepto arrangement, but Streptococcus is only one of the genera for which this arrangement is seen. Please take note that genera is a plural word for genus. So when we're talking about three different genera, we would say genera instead of genus. The next arrangement to look at is this packet of four. This is fittingly called a tetrad. So Micrococcus sometimes forms this tetrad. I used to also work with a bacterium called Pediococcus acetylestc. It was one of the lactic acid bacteria, and this bacterium also formed a tetrad. Now, lastly, the cocci can sometimes form packets of 8. These packets of 8 are a product of the fact that Sarcina arrangements the bacteria are dividing in 3 planes versus those that form tetrads were dividing in 2 planes, and of course the irregular planes of the grape-like cluster. So all of the arrangements are a product of how many planes the bacteria divide in. So looking now at the bacilli, so besides our good friend filbric, we also see other types of bacilli, and some bacilli are almost look as though they could be cocci. They're so short and fat that we call them coccobacilli. Now other rods, on the other hand, will sometimes bend and curve around, and they will form something that we call a Vibrio arrangement. Vibrio arrangement is also the name of a genus, so Vibrio fischeri is a bacterium that we will work with, but the ever famous Vibrio cholerae, which causes cholera, is another good example of a bacterium that forms a half-moon shape. Many photosynthetic bacteria are actually spiral cell shapes, and there are some awesome bacteria out there that are shaped just like corkscrews. For example, Treponema pallidum is a good example of a spirochete, and I am so fortunate to have a giant stuffed microbe, which visit me in my office sometime, and I will show you a lot of my giant stuffed microbes, but this is my little pal that is Treponema pallidum. Perhaps he needs a name too. I keep him on my lay just for safekeeping and sheer hilarity. But Treponema pallidum causes syphilis, the sexually transmitted disease. And notice that it does have that corkscrew shape. So if we follow this cell shape through, we'll see all the way that it is a corkscrew shape. Now, the way that Treponema pallidum moves is like a corkscrew. It's via a mechanism called an axial filament that allows it to literally move like a corkscrew. That gets it through super viscous solution. For example, for many free-living spirochetes live in mud, but we also see that they'll live in tissues, for example, the way that Treponema does. So there are other shapes that aren't listed here. There are bacteria that are square, that are cuboidal. We see bacteria that look like stars, and in fact they look like stars oftentimes because they have surface projections called prosthesia, and these prosthesia can sometimes be multiple and can make the bacterium change its shape literally. Now, there's actually a name for a bacterium that is a shape shifter. Now, not only did I think that Jeffrey was going to be super jazzed about all of the varying pathogens that we've just talked about, for example, Treponema pallidum, which has a lot of medical relevance, Staphylococcus aureus has a lot of medical relevance, of course, Streptococcus pyogenes that causes strep throat. But I also thought you would be super stoked to know that there are bacteria that are shape shifters. So it sounded to me like you liked science fiction a little bit, and science fiction kind of makes me it makes me think that this would be a character in a sci-fi, a shape-shifting bacterium. We call them pleomorphic. So pleomorphic bacteria shift their shape or change their shape. So some bacteria and archaeal cells have unusual shapes. I talked about squares. Some have surface extensions. I talked about prosthesia as those surface extensions. Some bacteria only have a single surface extension that allows them to reach out and attach themselves to certain surfaces. So these are the overview of shapes and arrangements. Let's talk about size. Bacteria range in their size. As a general rule, we could say that they're smaller than eukaryotic cells, but that's just a generalization because there certainly are some bacteria that are much larger than eukaryotic cells, and likewise there are some tiny eukaryotic organisms. This particular picture on the lower left is of something we've already introduced, Thiomargarita namibiensis. It was isolated off the coast of Namibia in the sandy soil, and you can see that it has internal sulfur storage granules, very fitting to the name, Thio. Namibiensis, of course, the species name elucidates the location that it was isolated. And the crazy thing about this bacterium is that it is huge, 750 micrometers. That is literally almost 1,000 times the size of some bacteria. So it is much larger. It is much larger than many eukaryotic organisms. So generalizations in size, bacteria ranging from 0.3 to 2 micrometers, you all will be getting an unknown in lab and you'll be estimating the size of that bacterium. However, we will find that most of your bacteria are on the order of maybe 1 to 5 micrometers, nothing like Thiomargarita namibiensis at 750 micrometers. Now, bacteria and archaea reproduce by binary fission. So I want to watch a short video that can be found on TeacherTube that shows how binary fission works. But the long of the short is that bacteria literally elongate and they double their contents, and then they split. They form a septum that cleaves them off into two distinct daughter cells. So from one mother cell cleaves to two daughter cells cleaves to two daughter cells, right? So each daughter cell forms two. Think about the exponential growth that happens there. So this short video, I'm going to narrate it for you. You can also find it on TeacherTube. So bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium dividing every 20 minutes could produce nearly 5,000 billion bacteria in one day. Whoa. That's why, like, when I think about people saying, nothing will change overnight, just sleep on it, don't worry about it, nothing will change. So wrong. If you've got a bacterial infection, when you think about the doubling rate of bacteria, a lot can change overnight, can't it? I like to say it's a lot like working with undergraduates, if there's some problem brewing, don't let it go overnight, because so much can change. Think of all the things that can change overnight. So this is all about binary fission. Let's go back to our good friend Philbrick, or Norbert, or whatever we're going to call him. And we're going to introduce some of the parts. So overarchingly, we can say that a bacterial cell is somewhat simpler in structure than the traditional eukaryotic cell, but of course we're going to find exceptions to every rule. Notice that within the bacterial cell, a sort of central region is shown. This is called the nucleoid. The nucleoid is a gel-like mass where the chromosome of the bacterium is stored. The chromosome of a bacterium is a single chromosome, a single circular piece of DNA. It's packaged with protein, and so this is kind of what this looks sort of like a spaghetti region in the center here of Norbert, but that's what that's showing. We can also see little pieces of nucleic acid, and in a little bit we're going to find out that those are called plasmids. Now we also have in here, and I don't know if you guys can see this very well, there's this white kind of globular structure. We're going to talk about something called inclusions, and inclusions are regions that can store or play a role in osmotic balance or buoyancy, and we'll be looking at those in detail. So we're going to be taking our time to zoom in and talk about all of the parts of a bacterial cell. We are actually going to start with the membrane. So notice this region bounding the cell and holding in its internal contents. I want to talk just a little bit about that region first. So that's where we'll go now.